You'd be like, "Hi, I'm Lee again," and then you'd be like, "Hi, I'm Christine such and such. We're science at the Danforth Center," and then like we're harvesting Setaria roots and shoots for our drought experiment. Here's how we're gonna do it.
Or we could just be like,
and then,
"My name's Max, and I'm photobombing this video."
and then like you have like the dry ice, and like so people on the internet when they see it, they'll be like, "Ah, yes, they put it in liquid nitrogen. That's what's cold,"
Or we could just have you do director's commentary.
No.
We will be comparing drought, well-watered and drought responses in A10, B100 root and shoot tissues across the 10-day time point. So this is just an example for one day. We will be harvesting a total of 32 samples, 16 for the shoot and 16 for root tissues. And we have them labeled 1 through 16. That includes A10, B100, well-watered, and drought. So what we're going to do is pre-label each of the tubes. We'll have a 13 mil tube and put 10 beads in there, put on the label sticker, and then also write down the date and the tissue type and number for each tube in case the labeling falls off. So I'm just going to show give an example of how to do this. First of all, we need to get 13 mil tubes. I'll put this away. You get 13 mil tubes, and these can be found in the Britnell lab, and also beads, which will be put in a bin, also in the Britnell lab. And we need to have labels, which will all be in the drought experiment bin. Grab a tube, and then also beads. Have a rack, it's easier. Count 10 beads. 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. Close the lid, and then get the label, which has the date, the session, A10 or B100, the tissue type and number, and also the treatment. So in this case, this is for October 10th, A10 root sample 1, and this is a well-watered sample. So put this onto tube, grab a Sharpie, and write down the date and the tissue type and number. So here, write down October 10th, and root number 1. And that's it.